सोममराति यतो निदहाति वेदः स नः पर्षदति दुर्गाणि विश्वा नावेव सिन्धुं दुरितात्यग्निः तामग्निवर्णां तपसा ज्वलन्तीं वैरोचनीं कर्मफलेषु जुष्टाम् दुर्गां देवीगुं शरणमहं प्रपद्ये सुतरसि तरसे नमः अग्ने त्वं पारयानव्यो अस्मान् स्वस्तिभिरति दुर्गाणि विश्वा ऊश्च पृथ्वी बहुला न उर्वी भवातोकायतनयाय संयोः विश्वानि नो दुर्गः जातवेदः सिन्धुन्न नावा दुरितातिपर्षे अग्ने अत्रिवन्मनसा गृहाणोऽस्माकं बोध्यविता तनूनाम् पृतराजितगुं सहमानमुग्रमग्निगुं हुवेम परमात् सदस्तात् स नः पर्षदति दुर्गाणि विश्वा क्षामद्देवो अतिदुरितात्यग्निः प्रत्नोषिकमेढ्यो अध्वरेषु सनाच्च होता नव्यश्च सत्से स्वाञ्चाग्ने तनुवं पिप्रयस्वास्मभ्यं च सौभगमायजस्व गोभिर्जुष्टमयुजो निषिक्तं तवेन्द्र विष्णोरनुसञ्चरेम नाकस्य पृष्ठमपि संवसानो वैष्णवीं लोक इह मादयन्ताम् कात्यायनाय विद्महे कन्याकुमारी धीमहि तन्नो दुर्गिः प्रचोदयात् ॐ शान्ति शान्ति शान्तिः।
Offering my humble pranams at Bhagavan's lotus feet, revered Vice Chancellor Professor Karani Sanjeevi, Chief Guest Professor V. Chandrasekhar, Center Director, Tata Institute of Fundamental Research, Hyderabad, Esteemed Director of the Campus, Dr. Raghavendra Raju, Dean of Sciences, Professor Sundareshan, Professor Janardhana Jagadishwar Rao, Heads of the Departments, invited speakers, delegates, my dear brothers and sisters, I extend a very warm welcome to each one of you to this 2-day symposium RACS 2021. Making science indeed is humanity's greatest wonder. We live in an era where yesterday's fiction becomes today's reality, and today's reality becomes history tomorrow as we all progress. The bedrock of research has always been refinement of error, but there is another important aspect, and that is a researcher's unshakable integrity. But that only leaves us with a greater role of promoting responsible imagination in today's young minds. With the opening of the world-class research center at the Sri Sathya Sai Institute of Higher Learning, there is not a more opportune stage and platform to promote inspired research even while being tethered to the character of humanity, to foster a temperament that combines science with society. This year, the theme we have chosen for the conference is titled Recent Advances in Chemical Sciences. The year bygone has been full of scientific surprises. Imagine my journey as an organic chemist for 30 years who woke to a news that carbon has been reported to form 5 and 6 bonds, and not to forget the ongoing global pandemic, where the barriers between biology, chemistry, and commerce have been dissolved to promote research that can help mitigate the world disaster caused by the novel COVID-19 virus. I am delighted to announce that from the Department of Chemistry, Sri Sathya Sai Institute of Fundamental Higher Learning, and the able leadership of Dr. R. Sai Satish, have come up with a prototype for COVID detection kit which is fast, portable and reliable. The kit is pending the final formalities required from the ICMR for its availability to the common people. Apart from this, other research pertaining to COVID-19 like application of medicinal herbs, drug repurposing, etc. have been undertaken. There is not a more important time to open our eyes to the world research and its perspectives, and therefore we wholly believe that this conference shall be a wonderful platform that complements the experience of the learned and the enthusiasm of the listeners. Every erudite speaker who consented to address this gathering of budding minds is a great researcher, scholarly men of incredible accomplishments, yet each one of them humble and kind. I am certain that in this interaction of our collective wisdom, we shall all grow together. In today's symposium, our respected Vice Chancellor, Professor Karani Sanjeevi, will be giving the inaugural address. It is my privilege to introduce him. Professor Karani Sanjeevi is the 11th Vice Chancellor of the Sri Sathya Sai Institute of Higher Learning. Professor Karani Sanjeevi, MD, PhD, is a professor at the Karolinska Institute, KI, in the Department of Medicine. Dr. Sanjeevi obtained his MD from Madras University, PhD from Karolinska Institute in 1994, and did postdoctoral work at the University of Washington in Seattle during 1994-95 and has been at KI till 2019. Dr. Sanjeevi heads a research group at the Center for Molecular Medicine in KI. He is editor for the Immunology of Diabetes series volumes published by the Annals of the New York Academy of Sciences and served in the editorial board of several journals like Diabetologia, Human Immunology, Current Diabetes Reviews, IELTS, etc. Dr. Sanjeevi also served in the Council of European Association for the Study of Diabetes, EASD, and had won several awards in the field of diabetes from EASD and diabetes research. He is visiting professor in several universities. He is also the secretary of the Immunology of Diabetes Society. Since 2000, he has served as KI scientific coordinator for India and promoted academy industry and industry partnerships between KI and India as well as Sweden and India. Since 2006, he has been Senior Advisor to Business Sweden, an agency that belongs to Ministry of Foreign Affairs, Government of Sweden. He is also Senior Advisor to the Innovation Office of KI and promotes ideas to IPs and finds Indian investment partners for such IPs. Dr. Karani Sanjeevi has been an independent director at Oncology Ventures ABRS since 2018. He is member of the scientific advisory boards of Diamid Medical and Seraxis. He is considered to be a global authority in diabetes, particularly autoimmune diabetes. He is a member of the Nobel Prize Selection Committee for Physiology and Medicine since 2015. He has several awards to his credit. I will just mention 3 prestigious awards which were conferred on him in the recent past. Dr. Karani Sanjeevi won Pravasi Bharatiya Samman Award, which is the highest award for person of Indian origin from Government of India. Dr. Sanjeevi received the award from the President of India in 2017. The award has been given to Dr. Sanjeevi for eminence in the field of one's field or outstanding work, which has enhanced India's privilege in the country of residence. Vice Chancellor Professor Dr. Sanjeevi has been chosen by the Scientific and Organizing Committee of NUCD, instituted by Konungo Institute of Diabetes Specialities Private Limited, which is a leading integrated diabetes and multi-speciality care provider in India, as this year's recipient of the Lifetime Achievement Award. It is a prestigious honor that serves to recognize the contribution made in the field of diabetic research. Vice Chancellor has been awarded Hello Diabetes Academia Hippocrates Oration in Academia 2020 Digital International Conclave, jointly conducted by Diabetes Care and Diabetes Foundation of India for the yeoman's service in the field of medicine and an exemplary delivery of oration on checkpoint inhibition therapy for cancer and development of type 1 diabetes mellitus. With these few words, I now request Professor Karani Sanjeevi to deliver the inaugural address. Saira.
My humble pranams at the lotus feet of Bhagwan. It gives me a great pleasure to be the opening batsman in this international symposium organized by the chemistry department called Recent Advances in Chemical Sciences. It's happening in 2021. Of course, 2020 had been a difficult year for everybody. And in this year, a lot of disruptions had taken place. With the result, a normal life could not be led by many people. This is because of the COVID pandemic, which the WHO declared as a pandemic in the month of March 2020. In this pandemic, lots of people have lost their lives. Economy has been disturbed. The problem that we are facing is insurmountable, and there are a lot of life lessons we have learned. In this COVID pandemic, the topic for my talk is going to be COVID-19 vaccine. When will the pandemic end? How will it end the pandemic? So my talk is going to give details about the vaccines, how much of the population needs to be vaccinated, and then when this vaccine will take effect that would prevent transmission from people to people, thereby arresting the spread of coronavirus, thereby the infection due to coronavirus. So it can happen in countries that are small in terms of population very quickly, whereas in a country like India, it is going to take a little longer time. And we haven't yet launched the vaccine in India yet. Hopefully, the vaccines are arriving, those that are made outside India. And India is also going to be a manufacturing hub for the vaccine, where quite a bit of world supply of the vaccine is going to originate from India. In addition to the foreign vaccines that are manufactured in India, primarily the Oxford AstraZeneca vaccine, there's also a company in the United States called Novavax. Their vaccine is manufactured in India. In addition to the two companies in India, one is Zydus Cadila, another is Bharat Biotech, who are bringing homegrown vaccine. And those, when they hit the market, it's going to accelerate the speed of vaccine delivery, vaccine inoculation, and initiation of immunity in the people who are vaccinated. Thereby, those vaccinated will no longer be able to spread the disease, so that we break the transmission of the disease. So my talk is going to be on COVID vaccine. How will it end the pandemic? And in this talk, I'm going to describe variety of situations, various vaccines, the doses in which you have to give them, the kind of complications you can expect, and how are we all going to come out of it very soon, most likely by the end of the year. The presentation that I'm going to give today is COVID-19 vaccine. How will it end the pandemic? So this is a presentation that is more for the scientific audience, but there's going to be a lot of information that the lay public is going to benefit. So what is COVID-19? COVID-19 is a disease caused by SARS-CoV-2, which is a respiratory virus, an RNA virus. And that is an updated version of earlier virus called COVID-1, which is also called SARS-CoV-1. So COVID-19 is a disease caused by SARS-CoV-2. In March 2020, WHO declared COVID-19 as a pandemic. So if you look at the picture, a scanning electron microscope picture on the left-hand side, you can see the COVID virus visible to you in orange, small spherical dots. And if you look at the picture below, as of December 31, 2020, you will see that there are 80 million people who are affected. And in European Union, 17,348,000 people have been affected by COVID. And what's frightening is that in the entire world, as of December 31, 1,770,695 people have died. And if you take Europe alone, it's 427,798 people. That includes United Kingdom. But if you look at India, the number of deaths in India compared to the rest of the world is much smaller. If you look at India's population, you will realize India is 1.3 billion, which is what? 1,300 million. And we have not even touched 1% of India's population in terms of the COVID infected people. So there are 99% of India's population that are uninfected. If that numbers have to be believed, this is a population that requires enormous planning and execution of the planning to make the vaccine available to all of them. So when you look at the COVID vaccine, why has it got such an attention compared to the previous version called SARS-CoV-1? The reason why it's got much greater attention is because it causes death. It causes death in those who are older individuals, and it causes death in the older individuals, especially if they have comorbidities like diabetes or hypertension or other respiratory diseases. So it is the death that makes people fear this virus. So we need to ensure that whatever we do, we try to prevent death from happening. So the patients usually present with fever, cough, shortness of breath, and tiredness. Diagnostics is important. It took some time for the government as well as the health administrators to come up with a plan on the best diagnostic kits that are available. The gold standard is RT-PCR. Then there are immunologically based assays called lateral flow assay, which is what is developed by SSIHL and undergoing validation. Is a test that would allow us to measure antigen and antibody, but at a fraction of a cost. $1 for antigen estimation and $1 for antibody estimation. And if you look at these two results together, you can come out with a very clear pattern on whether it is early infection or late infection or older infection or no infection. And over the period of time since March, India has experimented with a lot of generic drugs, which is hydroxychloroquine, ivermectin, and several antiviral combinations. And at SSIHL, we have done some wonderful work in terms of mathematical modeling to show which are all the antivirals when you give it in combination is most effective. And currently, we are also working on a very interesting project, which is, again, a mathematical modeling to tell you how many people should get vaccinated, whether one vaccination is enough or a combination of vaccines are best, in order to ensure that there is 100% efficacy for the vaccines that are given, so that people do not have to have any worry about getting reinfection. And most importantly, what is critical here is whoever is vaccinated should not be able to spread the virus to the next person. If that can happen with the vaccine, that is the greatest boon. That means you are breaking the spread of the COVID-19 from people to people. Social distancing, wearing masks, cleaning your hands regularly as hand hygiene. They are also measures, but if your immune system is primed to deal with coronavirus as it enters your body and does not allow the virus to infect your cells and multiply, you have achieved a great progress. That is going to break the transmission. The moment the transmission from individual to individual is broken, that's the end of the pandemic. Let's look at some of the details. If you look at the vaccines which can really be effective in stopping the pandemic on its tracks, you will realize that vaccines or a different cup of tea. In the world, people have developed variety of vaccines against a variety of infections, whether it is virus or bacterial. But sometimes you need to adopt some new technologies. And for COVID vaccines, there are nearly 274 vaccine candidates under development. All these vaccine candidates are in various stages of development. And in the last few weeks, 4 of those vaccines have been approved either by the United States FDA or United Kingdom's MHRA. And these vaccines have already started to be given to people, primarily those who are older individuals, and then coming down to healthcare workers and critical people who are exposed to the virus. When you look at the virus vaccine for COVID-19, you will realize that there are so many ways by which you can make COVID-19 vaccine. So one is a DNA vaccine, another is an inactivated vaccine, then you have a live attenuated vaccine, then you have a protein subunit vaccine, then you use virus-like particles which can encode or in which you can put some of the sequences that encode for the spike protein. So you're not having the full virus, but these virus-like particles will carry the sequence of some specific component, predominantly the spike protein component. And then there is another category called virus vectored. And then one more category, which is directly injecting the mRNA, since it is an mRNA vaccine. So many of these approaches are very interesting. Quite a bit of them have worked very well for other vaccines, other viral vaccines, whereas mRNA vaccine is the first time that it has been developed and used in humans. And this is really a breakthrough in terms of how it can prevent a viral infection from taking place. So we can look at all those things in the following slides. So when you look at the type of vaccines, you will see that you need to create as the active immunity and or you can create passive immunity. As you can see, you have DNA vaccine, which is developed by a company called Inaugural. You have RNA vaccine, which is developed by a company called Moderna and Pfizer. Both these have been approved in the United States and in UK, and in many countries are in the process of approving them. Then you have a viral vector vaccine, which is the Oxford AstraZeneca vaccine. And there is one more that has been approved in some places in the world, is Gamaleya Sputnik, which is the Russian vaccine. So we have Moderna vaccine, we have Pfizer vaccine, we have Oxford AstraZeneca vaccine, we have Gamaleya Sputnik vaccine. All of them have been approved. And Janssen's vaccine and Novavax vaccine, both are American companies, are awaiting. Hopefully in a month's time, these 2 vaccines should also become available. You also have vaccines that are developed by Indian companies, and they also fall into some of these categories, especially inactivated virus and live attenuated virus. Some of the companies are also producing what is called as passive immunity, where they generate the antibodies that are targeting the virus, and these antibodies go and neutralize the virus. When this happens, you realize that the antibodies will go neutralize the virus does not spread, and this is something that has been effectively used in individuals who have already developed infection, who are already in a critical stage, and in an intensive care setting, it has really brought benefit to a lot of people. So this is a very interesting approach, but it is called as passive immunity. But what we are trying to generate is the active immunity, that is to generate your own immune system to develop antibodies. And this kind of active immunity brought about by vaccination is far, far better than getting infected and recovered. When you get infected, there are a lot of side effects. And those side effects are many times damaging many organs in the body. That leads to lifelong disability. Whereas when you take these vaccines, it is not the whole virus that you're talking about, which means you will not have so many organ damage that can happen. And the antibodies that are developed have become robust, and they are called neutralizing antibodies that bind to the virus and prevents the virus from entering the cells where they can replicate. So when you look at the active immunity vaccines, you have nucleic acid vaccine, which is the DNA vaccine, RNA vaccine, or viral vector vaccines. These are the vaccines that have already been in the market, especially the RNA vaccine and viral vector vaccines. There are other vaccines that are under development. Hopefully in a couple of months' time, that should become available, which are the protein vaccines, where there is viral subunit vaccine, split virus vaccine, and all these vaccines have already been tested in other viral infections and have been working very well. So the model for preparation of these viruses are simplified in these cases, but all these vaccines need to go through an elaborate testing process, which is preclinical testing, clinical testing, before you can get the approval for launching these vaccines. So all of them are in different states of development, and hopefully as we progress in 2021, many of these vaccines will become available more and more, and these vaccines will start helping people to develop immunity against the COVID infection. So how does it all work? So if you look at the DNA vaccine, when you inject the viral DNA directly, it gets into the nucleus of the cell. It becomes RNA, then the RNA is transported to the cytoplasm where the proteins are made, and the proteins are released so that the immune system recognizes the proteins and starts developing immunity towards those proteins. So these proteins are predominantly spike proteins to which we are building immunity. And in the case of RNA vaccines, you directly inject the RNA. So you avoid the step of DNA going into nucleus, and the nucleus is trying to produce RNA and bringing the RNA into the cytoplasm. But you directly inject the RNA vaccines intramuscularly so that they can start making the proteins. And these proteins are the ones that serve as antigen for the immunity to develop and antibodies to develop to neutralize the virus. So there is one more vaccine called the protein vaccine, where you introduce the spike protein itself. That is a fully formed protein. So you do not know what the RNA vaccine is going to do, because this is the vaccine that has been developed for the first time. There is no precedence for this. But it is so far been the most effective, and I will look at some of the results. Protein vaccines are still not approved, and hopefully when that approved, it might appear simple. So when you give the dose, first dose, you get a primary immune response. And when you give the second dose, then you get a secondary immune response, which is much bigger than the primary immune response. And when you give the second dose and you develop the secondary immune response, you realize that you develop memory B cells and T cells. And these memory B cells and T cells provide you lasting immunity, so that even in future, when these these viruses are encountered by your body, these memory B cells and T cells should be effective in bringing out the immune response so that the viruses are eliminated very efficiently. So how does the RNA vaccine work? So RNA vaccine consists of an mRNA encoding the antigen, which is the antigen of interest is the spike protein. This is placed in lipid nanoparticle vehicle. The lipid nanoparticle prevent the mRNA degradation by the host until it is taken up by the cell. So vehicle is very important. Once administered, the RNA vaccine is taken up by the host. The intracellular lipases degrade the LNP, exposing the mRNA. The mRNA is then translated to spike protein, and it is on the cell surface, stimulating antibody response and T cell response. So this approach has been adopted by a company called Moderna, and another company called Pfizer, which is partnered with a small biotech company called BioNTech. This is actually been an effort that had primarily been used against cancer vaccines. And now in the pandemic, they have started using this against the coronavirus with very successful results. So both vaccines carry the foreign gene for the whole spike protein. So the spike protein is the primary protein that gives the antigenicity, leading to antibody formation. So you inject this vaccine intramuscularly, the vaccine enters the cells, and the same thing that I said in words are shown in pictures here, that prompts the immune system to produce antibodies, activate T cells, and destroy the infected cells. So this is what happens. And if the patient encounters the coronavirus, the antibodies and T cells are triggered to fight the virus so that the virus is completely eliminated even before it could infect a cell. That means you will not have any cell infected by this. So your lungs are definitely going to be protected, and you're going to be protected very well by this. So when you compare the COVID vaccine of Moderna and Pfizer that have already been approved, both have a lot of similarities, but the solution in which it has been suspended is different. The encasing of the RNA by fat, which is the lipid layer, is also different. But the method is more or less similar, and this method is the one that makes this vaccine at the end of the phase 3 clinical trial very effective. So when you look at some of the available vaccines that are already approved, you will see that Moderna and Pfizer vaccine has an efficacy of nearly 95%. This 95% means there are still 5% of individuals this vaccine is really not effective. But there are problems with the vaccine in terms of transportation, in terms of cryopreservation, and Pfizer vaccine requires a cold chain of minus 70 degrees, whereas the Moderna vaccine requires a cold chain of minus 20 degrees. And in an Indian setting, if these vaccines have to be taken to villages and rural areas, we don't have the cold chain established so that the vaccine could be made available to all the people in India. So the problem here is the vaccine, once it is taken out of that minus 70, within 30 minutes, it has to be reconstituted and then injected within a period of 3 hours. So this is a little bit difficult task to achieve in a rural setting. So we need to have other ways by which we should have the vaccine available. And in this case, the Oxford AstraZeneca vaccine is really, really beneficial. And all the approved vaccines have to be given in 2 doses. But the Oxford vaccine seems to have 62% efficacy, whereas that is after the first dose. When you give the second dose, after 4-week period, the efficacy improves only to 72%. Whereas if you give the vaccine instead of 4 weeks, little longer than 12 weeks, the efficacy improves much better. So when you look at these 2 important RNA vaccines, you will see that the storage is an important aspect. The transportation is an important aspect. But if you can do all these things, the vaccine is really very efficacious. And when you look at the side effects of the vaccine, these are the standard side effects that you will find on all the vaccines. But in the last 1 month, when the Pfizer vaccine has been introduced, people have started to see certain complications that you need to be aware of. In individuals who are at risk for developing anaphylaxis, which is the allergic reactions, whether it is to food allergy or drug allergy, these people have to be a little careful. What it means by careful is you need to have all the anti-allergy medications available, and in case you develop allergy, it should be possible for the doctors to treat these allergic reactions very effectively. If it is not well prepared, then the allergic reactions can lead to complications. So allergic reactions should not put people off from taking the vaccine. Whereas in United Kingdom, they have recommended for the Pfizer vaccine that it could be given to pregnant women and lactating mothers as well. So when you look at the efficacy of the vaccine, as I spoke about, there are varying levels of efficacy, but the lowest efficacy is only with regards to the Oxford vaccine. Then it comes to the question, in order to get 100% efficacy, can you combine these vaccines? It is possible because already University of Oxford AstraZeneca vaccine has started to partner with the Sputnik 5 vaccine. And they've already started a clinical trial by combining these vaccines and given. Hopefully, this should take care of the efficacy to reach 100% level. So such combinations are also going to be an important aspect as we move along in 2021 by giving these vaccines to people. So when you look at these vaccines, whether it's nucleic acid or protein, the administration has to be done by an injection, which is by intramuscular. The vaccine that is being developed in India by Bharat Biotech, the version of the vaccine can be given intranasally, which means there is much greater friendliness in terms of usage of the vaccine. So you need a syringe and a needle in order to vaccinate everybody. And that's a lot of syringe and needles that you'll have to have. And everybody should receive 2 doses. So if there are 1.3 billion Indians, you need to have 2.6 billion syringes in order to make the syringes and needles in order to make the vaccine effective. So from a logistic perspective, all these things need to be arranged by the government. So when you look at the safety and robustness of the immune response, all these vaccines, especially the RNA vaccines and viral vector vaccines, seems to have a very strong immune response. And this is the immune response that is very much needed in terms of neutralizing the virus if the virus enters the system. So can the vaccines end the pandemic? The answer is yes, of course, the vaccines will end the pandemic. And this is the paper that is published in New England Journal of Medicine on the Pfizer vaccine, where it clearly shows that the efficacy of the vaccine is 95%. And the 2-dose regimen of the vaccine confers protection in those 95% of individuals. And the population tested is above 16 years of age. And this is the paper that was published 2 days ago in New England Journal of Medicine. This vaccine is produced by the company called Moderna. And here also, the vaccine efficacy is 94.1%. And this has very transient, local, and systemic reactions as a side effect, and nothing that causes major illness. So these 2 vaccines are potentially safe. And when this starts rolling, the number of people who get vaccinated becomes very important in order to reach a stage where these coronavirus will stop spreading from people to people. So the question is, can the vaccines end the pandemic? Yes, of course. Can a single vaccine be enough? Maybe not, because none of the vaccines are giving 100% protection. So the efficacy rate is really not there. So can you combine a vaccine for 100% protection? Yes, it's possible. We need more studies. Already a study has started where they are combining the Sputnik vaccine with Oxford AstraZeneca vaccine. So when those results come out, maybe we need to give both the vaccines in order to get the level of protection. So there is an inflection point at which when you reach certain number of population with the vaccine, that is the point when the spreaders versus non-spreaders, the balance will shift, where there are too many non-spreaders who are vaccinated individuals, and they come across the coronavirus virus by itself, they will not be able to spread the virus to another person. So at some time point, you will suddenly see the transmission level dropping to very, very small numbers. More than the transmission, what concerns people is the number of deaths. And I hope in 5 to 6 months' time, the number of deaths should also drop to 0. In this regard, I would like to mention that in the mathematics department of SSIHL, we have already started modeling studies on when will the pandemic end. We should really feel proud that the whole scientific community have come together in this particular pandemic in order to bring the best for the people. And the government and all are working closely together so that the cost is not important, the profit is not important. Protecting people is the most important, especially protecting elderly, so that we can avoid death. Because death is the most important worry in this coronavirus pandemic. If you can prevent death, then it becomes like any other influenza infection that you can get through and get over. You have heard my talk on COVID-19 vaccine. How will it end the pandemic? And I concluded that the pandemic can be stopped by giving vaccination to as much people as possible. I'm sure that the whole population may not be willing to take the vaccine. There will be somebody who might not want the vaccine. But if you discount those people, if majority of the people take the vaccine is really much better than getting the virus and recovering from the virus, which might have some residual effects of the viral infection, which might not be there with the vaccine. even though the vaccine has mild complications like pain and fever few days after the vaccines and things like that, that is something that is not going to affect people in any way. The things that we'll have to wait and see is whether combination of vaccine is going to be effective, and if so, how quickly we give those vaccines, how quickly we cover the population who are vaccinated and make that number larger, so that in the end, we will hope that person to person transmission is broken. And at the same time, we protect the elderly so that they do not get infected and die. So if we can prevent death, that's a huge achievement. And if that is possible in 2021, I'm sure at the later part of the year or from 2022 onwards, we should be free from this virus and we can hopefully go back to the pre-pandemic time. So with this, I'll end my talk, and you have my email address here, and you can visit our SSIHL website to see all the work that we are doing in SSIHL with regards to COVID and other diseases. Thank you very much, and Saira.
I invoke Swami's blessings, love and grace. I have the privilege to introduce to the audience our distinguished guest, Professor Vadapalli Chandrasekhar. Professor Chandrasekhar obtained his MSc from Osmania University, Hyderabad, India in 1977. He received his PhD degree in 1982 from the Indian Institute of Science, Bangalore, under the supervision of Professor S.S. Krishnamurthy. He worked as a postdoctoral research associate with Professor R.R. Holmes at the University of Massachusetts, Amherst, USA, between 1983 and 1986, and then as a senior research officer in the Indian Petrochemicals Corporation at Vadodara for a year. He then joined the Department of Chemistry at the Indian Institute of Technology, Kanpur in 1987, where he has been a full professor since 1995. He served at the head of the Department of Chemistry, IIT Kanpur, between 2008 and 10, and a Dean of Faculty Affairs, IIT Kanpur, between 2011 and 12. He also worked at the Tata Institute of Fundamental Research, Center for Interdisciplinary Sciences, Hyderabad, as a senior professor, Dean, between 2012 and 14, and also as the director of National Institute of Science Education and Research, NISA, Bhubaneswar, India, between 2014 and 17. Currently, he is the Center Director of the Tata Institute of Fundamental Research, Hyderabad. His research interests are in the area of molecular materials, inorganic rings and polymers, main group organometallic chemistry, and polynuclear metal assemblies. He has been a recipient of several national and international awards, including the Shanti Swarup Bhatnagar Award of the Council and Scientific Industrial Research, India, and the Friedrich Wilhelm Bessel Research Award of the Alexander von Humboldt Foundation, Germany. He is an elected fellow of the Indian Academy of Sciences, Bangalore, and the National Academy of Sciences, Allahabad, the Indian National Science Academy, New Delhi, and the World Academy of Sciences, Trieste, Italy. He has been on the editorial board of several journals, including Organometallics. Currently, he is on the editorial board of Dalton Transactions as its associate editor. His research work is documented in 360 plus publications. May I request our distinguished chief guest to deliver his keynote address.
Sairam, thank you, and it's very nice to be on this conference on recent advances in chemical sciences. So So before I start, my pranams at Bhagwan's lotus feet, and I seek his blessings for this talk and for all our endeavors. I would like to acknowledge the various organizations, the Department of Science and Technology, and the various people who have collaborated with me, Professor Coliscio, Professor Clerac, Professor Jean-Pascal Sutter, Fabrice Poinchila, Dr. Rajaraman, and various students who have been involved in this work. So this talk is based on some of the work that we've been carrying out. This talk is going to be on single molecule magnets, which are compounds of exchange coupled clusters of paramagnetic metal ions. So these are molecular complexes where the transition metal ion or a lanthanide metal ion is encapsulated by an organic ligand. And these molecules, when you put them in a magnetic field, they retain their spin orientation even after switching off the magnetic field. So that is the important aspect in terms of at what temperature they retain their magnetization and how to ensure that this is retained for a very long time. And importantly, no intermolecular interaction is necessary for this phenomenon to occur. In fact, great lengths in terms of design is made to ensure that intermolecular interactions are prevented so that the entire magnetic response is obtained only from the molecule. Different types of single molecule magnets exist. This is a broad family. So among the various types of compounds that are present are, for example, the first of them is a manganese, large manganese ensemble containing 12 manganese ions, which are bridged to each other by oxide ligands. So this is a molecule where containing different large number of metal ions. You could have a chain like in a coordination polymer where you could have nickel 2 and manganese 3, for example, which are linked to each other by again bridged by different ligands. So in this particular case, so you have ligands that ensure that nickel 2 and manganese 3 have an electronic communication between them. Or in fact, you could just have one metal ion, like in this example where you have this ion which is encapsulated by an organic ligand. So any one of these systems where you have multiple metal ions or a chain of metal ions or just one particular metal ion can function provided we know what to do. So among the first examples is this manganese 12 compound which has two different types of manganese sites. You have a manganese plus 4 and a manganese plus 3. The manganese plus 4 is in the central location and the manganese plus 3 is in the periphery. The manganese plus 4 sites are among themselves ferromagnetically interacting, and the manganese plus 3 sites are also interacting ferromagnetically. Between them, their interaction is antiferromagnetic. So as a result of these interactions, the spin ground state is 10, and this gives rise to this phenomenon which was realized that if you have a large spin ground state accompanied by a magnetic anisotropy, then you can in fact harness this property. So as I was mentioning, the central cube which is present in this molecule is a cube containing manganese 4 plus ions, and it is surrounded by a ring of manganese 3 plus ions. And this combination generates this particular property. So there is ferromagnetic interaction between the set of manganese 4, there is ferromagnetic interaction between the set of manganese 3, and the two sets couple with each other antiferromagnetic. Okay. Now, as a result of several such studies done over the last perhaps a decade and a half, what is now realized is that the requirements for an SMM, for a synthetic chemist to target this, is that the electronic ground state should be bistable. There should be a large ground state spin, as I showed you, and there should be a uniaxial magnetoanisotropy, which is of the Ising type, that is, for example, preferably, let us say, the direction, and this gives rise to an energy barrier between the possible spin states. And this energy barrier, of course, then prevents a randomization or an equilibrium being reached. If the barrier is large enough, then the spins would stay put in one of the states. However, there are caveats that actually allow this ideal situation to be completely sort of obliterated by relaxation, and this can occur by quantum tunneling, which can, in fact, even at the ground states, even though there is a barrier, there can be an under barrier tunneling, which will result in equilibration. So this is, for example, shown over here. So when we start, let us say the spins are in the lowest spin states, either minus S or plus S state. You put the field on, then one of them is populated preferentially. And when you switch off the field, they're still populated. So ideally, if they have to only cross the barrier and before equilibration occurs, then you can make the barrier as high as possible and ensure that these spins are in the lowest possible state. However, because of various possibilities, these spins can readily go on to the other states that are there on the other side of the hill, and therefore fast relaxation occurs. So the whole process is how to prevent such relaxations and retain the magnetization in one of these wells. Given this, what are the possible candidates for single molecule magnets? So as I mentioned already, the first candidate that was actually looked at and from whose inspiration this entire field was born is this manganese 12 complex. So the idea was that if you can have a large number of transition metal ions put together in a molecule, which are paramagnetic and which can interact with each other so that a large magnetic movement can be realized, and if there is a magnetoanisotropy in such complexes, you possibly have a candidate for a molecular magnet. Additionally, you could also have a possibility where you can utilize two different types of metal ions. You could have a transition metal ion and a lanthanide. Both of them are put together in a complex and they can interact with each other, generating this property. Alternately, you also can have just lanthanide ions in a complex, and this also can give rise to this property. And in fact, all of these have been studied in great detail, and although my talk would focus on lanthanide ions, I would also allude to the other types of complexes. So in all of this, one takes the advantage of this very powerful technique of coordination chemistry, which is more than 100 years old, and in fact the Nobel Prize for coordination chemistry was as early as 1913 to Alfred Werner. But this technique still remains very, very active and alive, and can be utilized with great success for molecular materials. So what you're doing is you're designing ligands, interacting with metal precursors, and generating various molecular ensembles. Depending upon your design, you can utilize them for various purposes. So the way one can proceed in this area is, one, you do not know really what type of compound you want. You simply know that perhaps if I make a large cluster, that would be useful, but you really don't know how large the cluster should be, or in fact, how do you make them. So one of the ways you can do it is by this technique called serendipity. That is, you're realizing that since you really don't know what you want, but you simply have a hunch that if I have a large enough cluster, it should work, you're preparing a ligand that is multifunctional, and your metal and the ligand are interacting in such a manner that they leave unsaturation in terms of proliferation, and therefore you are likely to end up with a large cluster. The disadvantage of this method is, of course, that you don't know what the structure of the product is, and therefore, in fact, you probably have very little idea about what you're going to get, except that something interesting may happen. The advantage is that oftentimes the structure of the product is something that you would not have been able to design on pen and paper, and therefore you are just allowing nature to come up with a full sort of array of the nature's power and give you structures that you would not have thought about, and then analyze them at leisure. So several such compounds, for example, have been prepared. In fact, the first compound which I talked about, the manganese 12, itself has been realized by such a method. So which was prepared by manganese acetate and acetic acid and potassium permanganate. Clearly, they had absolutely no idea that you were going to get a manganese 12 compound, but this is what they obtained, and which actually opened up the field of single molecule magnetism. And in fact, even in our research unrelated to this field, where we've been working on multifunctional ligands, we have encountered such types of very interesting results. As you can see here, a copper 26 lipophilic copper 26 complex soluble in organic solvents, bridged together by a phosphonate ligand. And in fact, several lanthanide macrocycles that we could prepare by utilizing similar technique. But in all of this, we just did not have an idea that this was the compound that we are going to generate. It was only after the reaction was concluded and the reaction was worked up that we were able to isolate such products. And all of this was accomplished by using polyfunctional ligand. So essentially, the feature of the ligand is they have multiple coordination sites. They have features like, for example, keto-enol tautomerism, as present over here. There is flexibility of the ligand by, for example, rotation around the NN bond, and so on. And there are points like this phenolic unit which can bridge different metal centers. On the other hand, while this method of utilizing these polyfunctional ligands is very good and can give you compounds which you could not have perhaps anticipated or even designed, it suffers from the drawback that you can't test a hypothesis. Let us say you want to test a hypothesis that if I want to have a cobalt and let us say a terbium in a complex, and they are separated or they are bridged by an oxide ligand, what would be the property? So this is something that you can't test by the serendipity based method. But on the other hand, you can do this by a technique which is the design synthesis. So here you are designing a ligand which is specifically interacting with a metal ion and specifically giving you a compound whose structure can be predicted a priori. The disadvantage is, of course, that you are limited by your imagination and you are not using the full potential of the nature. But the advantage is you can make a compound whose design is predictable and you can test various hypotheses. So with this sort of a background, we started to look at various ligands, which could be called as compartmental ligands, where we have different compartments that can encapsulate different metal ions. So for our purpose, let us say we have a compartment that can specifically bind to a transition metal ion, and another compartment that can bind specifically to a lanthanide metal ion, and then see what happens. And since we know clearly what is happening, it is possible to systematically do this particular study and engage in this study and get to know what's happening. So we looked at phosphorus ligand, and this phosphorus ligand has different coordination sites, but in spite of the fact that it is polyfunctional, it is really specific. The inner pocket is specific to transition metal and the outer pocket is specific to lanthanides. So, which is, for example, has this methoxy donor, which is not a very good donor for a transition metal, but a very good donor for lanthanide. So, using such a concept, we are able to prepare compounds which contained in the center a lanthanide and on either side of it transition metal, and the connection between the transition metal and the lanthanide is through these oxide bridges or the phenolate. So we know that this is specifically prepared like custom ordered, if you want to say, prepared in this particular manner by use of this ligand, and then we can study the property knowing that the lanthanide and the transition metal are bound to each other, bridged to each other by this oxide ligand. And of course, we can, after all the analytical studies, including structure solution by crystallography, which tells you what the structure is, like this gadolinium has an icosahedral geometry around it, the cobalt has a distorted octahedral geometry around it, and the connection between them is through phenolates. We can study them, and we found out that these are excellent single molecule magnets. So once we knew this particular technique of doing this, we of course applied it to various other scaffolds. So here are examples where we utilized it on a ferrocene scaffold. Again, building a compartmental ligand, which is got one compartment that is specific for transition metal, another compartment specific for lanthanide, and we could prepare several such compounds. Where we had here in this example that I'm showing you, you had nickel and lanthanide connected to each other by the phenolates and also connected to each other by the acetate. Now, with this sort of a background, let me just take you to much more recent work, where we use this technique of preparing a compound where the entire magnetism is going to come through only one metal ion, namely in this case cobalt-2, because yttrium-3 is a diamagnetic species and won't contribute to the magnetism. So the synthetic technique is very similar. You have one pocket which is specific to transition metal, the other pocket specific to the lanthanide metal ion, and by utilizing this sequential sort of protocol of addition, you can specifically generate compounds in almost quantitative yields where you have cobalt and yttrium located in this manner and connected to each other by the phenolates. So these compounds which are connected have of course their structures were solved by using X-ray crystallography. So the main feature is to show you that the cobalt is has a distorted octahedral geometry, and the yttrium As you can see here, there are several coordination sites, and these are filled up by oxygens, and you have 9 coordination around yttrium. We have a candidate that would behave like a single molecule magnet. And how do you know that these molecules have this behavior? So one of the ways one can do this is by doing magnetic measurements, but utilizing AC measurements, AC magnetic measurements. So in the AC magnetic measurements, for example, in this plot that I'm showing you, you have this in the plot of chi double prime m versus nu, or chi double prime m versus temperature, you have this maxima that are generated, which depend upon the frequency of the measurement. And the reason this happens is because if you had a normal paramagnetic substance and you were to vary the frequency of the AC field quite rapidly also, you would not find any difference, because a simple paramagnetic substance can adjust to this variation without any difficulty. But if you have a magnet that is getting magnetized, then there is a lag time during this process, and that is what you pick up by this measurement, and that tells you that the system is magnetized, and therefore the speed of the frequency at which you vary the field has a direct contribution in terms of what you are seeing. So with this sort of a knowledge that just one particular ion, in our case the cobalt-2 itself, can contribute to magnetism, we were looking at single ion magnets based on lanthanide ions. So our aim was to prepare compound that had specifically a lanthanide in a particular geometry, and the geometry we were looking at was pentagonal bipyramid geometry. And so we wanted to fix lanthanide in this geometry, and so we had to design a ligand which would yield such a geometry. So a ligand, therefore, if you can just see the synthesis, starts off with this multifunctional sort of approach, where your final ligand has several nitrogens and has also this oxygens that are present over here, with the possibility that in fact the oxygen can be in a ketoenol tautomerism with the adjacent nitrogen over here. So we anticipated that here you have a large enough pocket that can accommodate the lanthanide ion, and in fact that would have the lanthanide ion in the plane of this pocket, and perhaps you could have the axial sites that would be free for other coordination. And in fact this works like this. So we have this pocket which is interacting with the lanthanide, and you have the axial sites over here occupied by the chlorides, and this compound was studied on its own as well as by dilution with a diamagnetic host and studied, and both of these studies gave very good results in terms of their single molecule magnet property. So here is just to show you that this geometry is nearly perfectly pentagonal bipyramid, with the axial sites having the chlorides and the equatorial pentagonal plane occupied by the various nitrogen and the oxide ligands that the ligand provides. And this can in fact be duplicated with other lanthanide ions, like for example the terbium over here. And you can see the very nice plots of the AC susceptibility versus temperature, and similar things you can also see with the chi double prime versus the nu plots. Now one other small detail in this type of studies is there is always this question, are these properties that you are generating, is it a result of intermolecular interactions, or is it just because of the molecule? So one of the ways of resolving this is by actually diluting it in a diamagnetic host which is completely isostructural to the parent that you're looking at. So we prepared an yttrium complex which is also pentagonal bipyramid, co-crystallized it with the dysprosium in a ratio of 10:1. So you have a diamagnetic host which is hosting the dysprosium guest, and therefore the statistical probability that two dysprosium molecules is minimized, and you repeat the study. And when you repeat the study, if it gets very similar properties, then you know that the molecular property that you're seeing is completely from the molecule and not as a result of intermolecular interactions. And therefore, as a result of these studies, when we get this and we analyze this, we found that the energy barrier is about 70 K, which is reasonable but not very high. So we thought, is it possible for us to increase the crystal field strength in the axial site? And we did this by putting a phosphine oxide, a tricyclohexyl phosphine oxide, and one chloride is still retained. And you can see really a dramatic increase in the energy barrier, which goes nearly threefold from 70 to 204. And in fact, all other properties are also really matching with this anticipation. So this kind of study, which is the design-based study, therefore allows us to test this hypothesis. We knew that increasing the axial crystal field would have a beneficial effect in this property, because we knew that this increasing the axial crystal field would actually increase the spacing between the MgS states of the lanthanide ions, between the ground and the excited state, and that should result in improved properties. So this is one of the benefits of doing the studies in a systematic manner, so that you analyze the result, come to a hypothesis, and that hypothesis can be further verified by a new experiment. So this was a result that was published very recently. Similarly, we had, having known that this pentagonal bipyramid type of geometries are quite important in these kinds of studies, we started to see if there are other ways we can generate them. And this is again two further papers that have just been published from our group. So one of them deals with a sterically hindered acetyl acetonate ligand. So you can see that there is a mesetyl group over here on either side. So normally you have this methyl acetyl acetonate, which is the methyl group. This has been replaced with a mesetyl group on either side. And when we interact this with the lanthanide salt, we have distorted 7-coordinate geometry arising at this lanthanide. The 7th coordination being taken up by the DMF molecule, which also, like in the previous examples, this compound shows very good behavior. Similarly, when we use a multifunctional ligand, such as what I've shown over here, and again by design, which probably is not so obvious, but maybe I'll just allude to it, you have this phenolate which can bridge two different metal centers, and then you have the possibility of either a keto or an enol tautomerism, which would depend upon the base that we use in the reaction medium. So if you minimize the base, you can have the keto group interacting, and that's what happens over here with the H, for example, not getting deprotonated, and we generate a dinuclear complex, both of the dysprosium being in pentagonal bipyramidal geometries. And both these compounds show very good SMM properties, as I had shown earlier. So I think basically what I wanted to show you in this very short talk was where is this field? This molecule, single molecule magnets was an area that was started with purely academic interest, where molecules which could behave like conventional magnets was discovered, and several approaches of preparing them are now becoming more and more apparent about what are the factors that needed to be modulated to give you better and better magnets. And once we know that, I think we are in a situation to harness the properties and see if we can get them into some applications. To summarize, therefore, what I've shown you is a variety of synthetic strategies for 3D, 4F, and 4F complexes. Some of them are based on what could be called as serendipitous methods, but many of them are based completely on design. And the trend is currently to move away from the serendipitous based methods to more towards rational design. One of the topics, I did not really dwell much on this, is it is becoming more important that it is not just enough to have a large ground state spin, but it is very important to have significant magnetic anisotropy that can be brought in either structurally or because of the inherent nature of the metal ion that is involved, and that contributes to the overall property. And finally, mononuclear single molecule magnets or the single ion magnets seem to be holding a lot of promise, at least in terms of understanding this phenomenon, because the number of variables get reduced. You have just one paramagnetic metal ion, and what you're really doing is change the ligand environment around it, so it allows you to understand what's happening really well. So with that, Sai Ram again, and thank you very much for this opportunity.
I'm extremely thankful to Professor B. Chandrasekhar for his enlightening presentation on the lanthanide complexes as single ion magnets. I'm sure all the young researchers will start working on this exciting field. It's given very valuable inputs. I, being an organic chemist, I'm also excited as I saw many of the organic molecules being used as ligands for this single molecule magnets. I'm very thankful, sir. In spite of your very busy schedule, you have made it possible to address the audience. I'm extremely thankful for your presence today. Sairam.
Saira.